Role of enzymes. Introduction. Plant enzymes play an important role during diseases. Cell walls contain proteins and enzymes that actively work to reshape the wall during cell growth and also thicken and strengthen the wall during induced defense. When a plant cell detects the presence of a potential pathogen, enzymes catalyze an oxidative burst that produces highly reactive oxygen molecules capable of damaging the cells of invading organisms. Reactive oxygen molecules also help strengthen the cell wall by catalyzing cross linkages between cell wall polymers, and they serve as a signal to neighboring cells about the attack. Plant cells also respond to microbial attack by rapidly synthesizing and depositing callose between the cell wall and cell membrane adjacent to the invading pathogen. Callose deposits, called papillae, are polysaccharide polymers that impede cellular penetration at the site of infection, and these are often produced as part of the induced basal defense response. Cutinases, cellulases, pectinases and ligninases are often secreted by the pathogenic organism. Fungi, nematodes and bacteria are all known to produce one or more of the above enzymes in specific pathogen-host combinations. Viruses and viroids generally do not secrete enzymes, although some viruses may encapsulate an enzyme in their particle. Pathogenic organisms either continually secrete enzymes or upon contact with the host plant. The first surface of an organism comes into contact with its cuticle and the cell wall of the plant. The cuticle is comprised of a complex wax cutin which impregnates the cellulose wall. The cell wall is composed of cellulose, which constitutes for the structural framework of the wall, along with the matrix molecules hemicellulose, glycoproteins, pectin and lignin. Thus, penetration into living parenchymatous tissue and degradation of middle lamella is due to the action of one or more enzymes which degrade these chemical substances. Cutinases degrade the cutin on the cuticle layer, pre-softening the tissue for mechanical penetration or as a first step in tissue degradation. Studies have shown that several fungi and at least one bacterial species produce cutinases. Also, some studies indicate that cutinases are continually produced in low concentrations, with degradation products often inducing even higher levels of cutinase secretion. In some organisms, cutinase production may also be linked to virulence. Peptic substances comprise the middle lamella and also form an amorphous gel between the cellulose microfibrils in the primary cell wall. Pectin degrading substances, often termed pectinases or pectolytic enzymes, include pectin methyl esterases, which is in short called PME, polygalacturonases, which in short is called PG, and pectin lysis or transaminases. Pectin methyl esterases remove small groups such as methyl groups, often altering solubility and thus affecting the rate of chain splitting by polygalacturonase and pectin lysis. Polygalacturonases also split chains by adding a molecule of water, while pectin lyases split chains by removing a water molecule from the linkage. Pectin degrading enzymes are involved in a wide range of plant diseases, particularly in the soft rot diseases. Examples are bacterial soft rot due to Irvina carotovora, leaf drop of lettuce and water soft rot of crucifers, and damping off in various seedlings due to rhizoctonia. Hydrolytic enzymes are produced by some plants in response to pathogens and often accumulate in extracellular spaces where they degrade the cell walls of pathogenic fungi. Chitinases are enzymes that catalyze the degradation of chitin, a polymer which is similar to cellulose that is present in the cell walls of true fungi. Glucanases are enzymes that catalyze the degradation of glycosidic linkages in glucans, a class of polymers similar to cellulose that is present in the cell walls of many oomycetes or water moles. In vitro analysis has verified the antifungal properties of these compounds, and transgenic plants expressing high levels of these enzymes exhibit increased resistance to a wide range of both foliar and root pathogens. Lysozymes are hydrolytic enzymes that are capable of degrading bacterial cell walls. Let us now study cellulases. Cellulose is the major framework molecule of the plant cell wall, existing as microfibrils with matrix molecules, which is glycoproteins, hemicellulases, pectins, lignins. They fill the spaces between the microfibrils and cellulose chains. Cellulases have been shown to be produced by many pathogenic fungi, bacteria and nematodes. Cellulolytic enzymes play a role in softening and disintegration of cell walls. No doubt, cellulolytic enzymes are involved in the invasion and spread of the pathogen, but also are instrumental in the collapse of cells and tissues. Cellulolytic enzymes participate indirectly in disease development by releasing soluble sugars that may be used as nutrients by pathogens, and also may be involved in the release of materials in the vascular system, interfering with transport or translocation of water. There are 5 general types of cellulases based on the type of reaction catalyzed. They are endocellulases. This enzyme randomly cleaves internal bonds at amorphous sites that create new chain ends. Exocellulase. This enzyme reacts by cleaving 2 or 4 units from the ends of the exposed chains produced by endocellulase, resulting in the tetrasaccharides or disaccharides such as cellulobiose. There are 2 main types of exocelluloses or cellulobiohydrolases. CBHI, which is called in short form. It works processively from the reducing end, and CBHI works from the non-reducing end of cellulose, and CBH2 works from the non-reducing end of cellulose. Cellulobiase or beta glucosidase. This hydrolyzes the exocellulase product into individual monosaccharides. Oxidative cellulases. This plays a role in depolymerization of cellulose by radical reactions, as for instance cellulobiose dehydrogenase. Cellulose phosphorylases. This plays a role in the depolymerization of cellulose using phosphates instead of water. In case of cellulase activity, the enzyme complex breaks down cellulose to beta glucose. This type of cellulase is produced mainly by symbiotic bacteria in the ruminating chambers of herbivorous. Aside from ruminants, most animals, including humans, do not produce cellulase in their bodies and can only partially break down cellulose through fermentation, limiting their ability to use energy in fibrous plant material. Enzymes that hydrolyze hemicellulose are usually referred to as hemicellulase and are usually classified under cellulase in general. Enzymes that cleave lignin are occasionally classified as cellulase, but this is usually considered erroneous. Within the above types, there are also progressive and non-progressive types. Progressive cellulase will continue to interact with a single polysaccharide strand, and non-progressive cellulase will interact once, then disengage and engage another polysaccharide strand. Most fungal cellulases have a two-domain structure with one catalytic domain and one cellulose binding domain that are connected by a flexible linker. This structure is adapted for working on an insoluble substrate, and it allows the enzyme to diffuse two-dimensionally on a surface in a caterpillar-like fashion. But there are also cellulases, mostly endoglucanases, that lack cellulose binding domains. These enzymes might have a swelling function. In most of the bacteria, cellulases in vivo is a complex enzyme structure organized in a supramolecular complex, the cellulosome. They contain roughly 5 different enzymatic subunits representing namely endocellulases, exocellulases, coelobiosis, oxidative cellulases and cellulose phosphorylases, wherein only endocellulases and coelobiosis participate in a hydrolysis. Molecular biology of cellulosomes had led to the discovery of numerous cellulosome-related signature sequences known as doctrines and cohesins. Depending on their amino acid sequence and tertiary structures, cellulases are divided into clans and families. Let us now look at hemicellulases. Hemicelluloses are complex polysaccharide polymers that link the ends of pectic compounds to cellulose microfibrils. Since hemicelluloses are such a diverse group of polymers such as xyloglucans, glucomanins, galactomanins, arabinoglucans, etc., several hemicellulases have been identified in many plant pathogenic fungi. The mechanism by which they participate in cell wall breakdown is not clear, nor is known how they contribute to pathogenesis. A hemicellulose, also known as a polyiose, is one among several heteropolymers such as arabinoxylans present along with cellulose in almost all plant cell walls. While cellulose is crystalline, strong and resistant to hydrolysis, hemicellulose has a random amorphous structure with little strength. It is easily hydrolyzed by dilute acid or base as well as a mydriad hemicellulose enzyme. Hemicelluloses include xylin, arabinoxylin, glucomannan and xyloglucan. Hemicelluloses are embedded in the cell walls of plants, sometimes in chains that form a ground. They bind with pectin to cellulose to form a network of cross-linked fibers. Hemicellulose found in hardwood trees is predominantly xylin with some glucomannan, while in softwoods it is mainly rich in galactoglucomannan and contains only a small amount of xylin. The average molecular weight is lower than that of cellulose at less than 30,000 as opposed to the 1 lakh average molecular weight reported for cellulose. Let us now look at ligninases. Lignin is a phenylopropanoid which is found in the middle lamella and secondary cell wall of plants. Lignin confers the tough, woody nature to woody tissues, and only about 500 species of fungi are capable of decomposing wood. Most lignin degradation is by Basidiomycetes, known as white rot fungi. These fungi produce ligninases that enable the fungi to utilize lignin. Ligninase is the original term encompassing many different types of oxidative extracellular fungal enzymes which catalyze the breakdown of lignin, which is commonly found in the cell walls of plants. Instead of the term ligninase, the term lignin modifying enzyme can also be used, since these enzymes are not hydrolytic but oxidative by their enzymatic mechanisms. LMEs include perioxidases such as lignin peroxidase, manganese peroxidase, and versatile peroxidase, and many phenyl oxidases of lactase type. LMEs have been known to be produced by many species of so-called white rot basidiomycetes fungi. They are produced not only by wood white rotting fungi but also by litter decomposing basidiomycetes fungi such as Agaricus bisporus and many copreous and agrobee species. The brown rot fungi, which are able to colonize wood by degrading cellulose, are not able to produce LMEs. However, efficient lignin and lignin-like polymer degradation is only achieved by fungal LME peroxidases and lacases in combinations with organic change transfer mediator compounds. Lacases are more widely distributed enzymes belonging to the multi-copper oxidase superfamily encompassing all three domains of life: bacteria, archaea and eukarya. Lipids and lipid metabolites influence pathogenesis and resistance mechanisms associated with plant microbe interaction. Some microorganisms sense their presence on a host by perceiving plant surface waxes, whereas others produce toxins that target plant lipid metabolism. In contrast, plants have evolved to recognize microbial lipopolysaccharides, sphingolipids, and lipid binding proteins as elicitors of defense response. Studies have demonstrated that the plasma membrane provides a surface on which some plant resistant proteins perceive pathogen derived effectors and thus confer race specific resistance. Plant cell membranes also serve as reservoirs from which biologically active lipids and precursors of oxidized lipids are released. Some of these oxylipins, for example jasmonic acid, are important signal molecules in plant defense. Arabidopsis thaliana is an excellent model plant to elucidate the biosynthesis and metabolism of lipids and lipid metabolites and the characterization of signaling mechanisms involved in the modulation of plant defense responses by phytolipids. Pectinase. This is an enzyme that breaks down pectin, a polysaccharide found in plant cell walls. Commonly referred to as pectic enzymes, they include pectolase, pectozyme, and polygalactoronase. One of the most studied and widely used commercial pectinases is polygalactoronase. It is useful because pectin is the jelly-like matrix which helps cement plant cells together. Other cell wall components such as cellulose fibrils are also embedded. Therefore, pectinase enzymes are commonly used in processing involving the degradation of plant material, such as speeding up the extraction of fruit juice from fruit, including apples. Pectinases have also been used in wine production since the 1960s. The function of pectinase in brewing is twofold. First, it helps in breakdown of the plant material, typically the fruit material, and also helps the extraction of flavors from the mash. Secondly, the presence of pectin in finished wine causes a haze or slight cloudiness. Pectinase is used to break this down and so clear the wine. Pectinases are enzymes that break down pectic substances in plants. Pectic substances are integral in maintaining the order of cells in plant tissue. When pectic chains are broken down, cells lose strength and shift within the walls of a plant such that the plant no longer has a rigid structure. This leads to wilting of varying degrees. Diseases such as fusarium wilt and verticillium wilt, which affect crops like tomato, potato, pepper and eggplant, produce enzymes that break down pectic chains, liquefy pectic substances and weaken the cell walls. Pectin degrading enzymes are also involved in the development of plant diseases that lead to soft rotting of plant tissue. They can be extracted from fungi such as Aspergillus niger. The fungus produces these enzymes to break down the middle lamella in plants so that it can extract nutrients from the plant tissues and insert the fungal hyphae. If pectinase is boiled, it is denatured, thus making it harder to connect with the protein at its active site and produce as much juice. Pectinases are also used for retting. Addition of chelating agents or pre-treatment of the plant material with acid to enhance the effect of the enzyme. As with all enzymes, pectinases has an optimum temperature and pH at which they are most active. For example, a commercial pectinase might typically be activated at 45 to 55 degrees centigrade and work well at a pH of 4.5 to 5.5. Pectinase is commonly used in fruit industries to speed up fruit juice extraction. The next enzyme, protease. This is also termed peptidase or proteinase. This is an enzyme that performs proteolysis, that is, begins protein catabolism by hydrolysis of the peptide bonds that link amino acids together in the polypeptide chain forming the protein. Proteases have evolved multiple times and different classes of proteases can perform the same reaction by completely different catalytic mechanisms. Proteases can be found in animals, plants, bacteria, archaea and viruses. Plant genomes encode hundreds of proteases, largely of unknown function. Those with known function are largely involved in developmental regulation. Plant proteases also play a role in regulation of photosynthesis. Vascular processing enzyme. In short, it's called as the VPE. Vascular processing enzyme would determine to play important roles in various types of cell death in plants. Early studies demonstrated that the identification of VPE as a vascular hydrolytic protein responsible for maturation of vascular proteins. Later, Nicotiana benthamiana VPE was reported to mediate virus-induced hypersensitive response by regulating membrane collapse. The next is cutinase. This is an enzyme that catalyzes the chemical reaction. Cutinases are enzymes that enable fungi to grow on plants by breaking down cuticular wax. Plants produce cuticular wax as a means of protection. The substance prohibits foreign agents from growing on and penetrating the exterior of the plant. Diseases like Puccinia hordi, which spreads destructive rust spores across the surface of plants, produce cutinases so that these spores are able to take root. The enzyme destroys cuticular wax by catalyzing chemical reactions that will break down the molecular components of the substance. This enzyme belongs to the family of hydrolases, specifically those acting on carboxylic ester bonds. The systematic name of this enzyme class is cutin hydrolase. Aerial plant organs are protected by a cuticle composed of an insoluble polymeric structural compound, cutin, which is a polyester composed of hydroxy and hydroxy-epoxy fatty acids. Plant pathogenic fungi produce extracellular degradative enzymes that play an important role in pathogenesis. They include cutinase, which hydrolyzes cutin, facilitating fungus penetration through the cuticle. Inhibition of the enzyme can prevent fungal infection through intact cuticles. Cutin monomers released from the cuticle by small amounts of cutinase on fungal spore surfaces can greatly increase the amount of cutinase secreted by the spore, the mechanism for which process is yet unknown. Cutinase is a serine esterase. The protein belongs to a alpha-beta class with a central beta sheet of 5 parallel strands covered by 5 helices on either side of the sheet. The active side cleft is partly covered by 2 thin bridges formed by amino acid side chains, by contrast with the hydrophobic lid processed by other lipases. The protein also contains 2 disulfide bridges which are essential for activity, their cleavage resulting in complete loss of enzymatic activity. Plant cell walls are heterogeneous and dynamic structures composed of polysaccharides, proteins and aromatic polymers. Cell wall composition and structures differ among plant lineages. The cell walls of angiosperms, which are the flowering plants, and gymnosperms, including conifers, all contain cellulose microfibrils embedded in a matrix of pectin, hemicellulose, lignin and structural proteins. But the type and relative amounts of these structural polymers differ among groups of plants and also change as the wall matures. For example, the walls of poales, grasses and other common linoid monocots differ from dicots and non-common linoid monocots in several ways. Type 1 cell walls found in non-common linoid monocots and dicots are generally rich in xyloglucan and pectin. In contrast, the type 2 cell walls of common linoid monocots contain glucuronoarabinoxylin as a major non-cellulosic polysaccharide. Common linoid monocots are also unique in having mixed linkages beta 1, 3, 1, 4 glucans. There are additional differences in the types of lignin and ferulic acid esterification found in grasses and dicots. In addition to the physical complexity of the cell wall, it is a dynamic structure that changes as the plant grows and ages. During cell wall maturation, from a primary to secondary wall in both monocots and dicots, the amounts of xyloglucans, pectins and structural proteins decrease while the amount of xylins and lignin increase. The major constituents of typical secondary cell walls are cellulose 35 to 45% dry weight in grasses, 45 to 50% in dicots. The second is xylins, which is 40 to 50% in grasses and around 20% to 30% in dicots, and lignin, which is 20% in grasses and 7 to 10% in dicots. Hence, to conclude with, these enzymes play a very significant role in plant pathology. Thank you.